Hello, my name is Wang. I'm currently a senior at the university school. Today, I'm going to introduce my independent research project, the artificial evolution of hydrogenase using gaming GPUs. Human demand for energy has reached an all-time high. Currently, more than half of our energy comes from fossil fuels. This consumption of energy has had a major impact on the Earth, including global warming, rising sea levels, changes in agriculture, increases in severe weather, and the spread of tropical diseases. Scientists agree that we are on a path toward disaster, and the only way to reduce the frequency of these disasters is to transition away from our dependence on fossil fuels. As we know so far, hydrogen energy is one of the most promising new energy sources. It is clean because the only product of combustion is simply water. At the same time, it also has a high energy density, which allows it to have a longer duration and high conversion efficiency. While the potential of hydrogen energy is widely recognized, there is still a question of how to efficiently produce it. Currently, the main method of producing hydrogen energy is through the electrolysis of water. However, these methods have technical limitations when it comes to large-scale production of hydrogen. Biological hydrogen production has gained attention for its eco-friendliness and cost-effectiveness, primarily relying on hydrogenase enzyme to catalyze hydrogen formation. Enhancing the efficiency of this enzyme is crucial for multiple reasons. It lowers production costs, it boosts environmental benefits, and improves scalability. Efficient hydrogenase enzymes speed up hydrogen production rates and often exhibit greater stability under varying conditions, thereby becoming more reliable for industrial application. High energy efficiency also leads to fewer byproducts, minimizing the need for additional purification steps. One of the most efficient biological hydrogen production methods is the use of algae streams that directly use solar energy to produce hydrogen. The key to inducing hydrogen production is through the use of an enzyme called hydrogenase. Hydrogenases have an efficient proton transfer pathway that is crucial for their function. This pathway shuttles protons from bulk water into the catalytic center of substrate or release product from the catalytic center into the bulk water. Understanding the catalytic mechanism of hydrogenase is fundamental. The enzyme contains a metallocluster active site, often with iron nickel, that facilitates a series of steps: electron uptake, proton binding, H bond formation, and product release. Insights into this mechanism are vital for bioengineering improvements in enzyme efficiency, stability, and tolerance to varying conditions. In the previous study, I utilized gaming GPU cards to conduct simulations that explored the enzyme's activity, specifically focusing on how the properties of amino acid residues along the proton transfer pathway impact enzymatic function. My simulation data indicates that mutations within this proton transfer pathway have the potential to influence the enzyme activity by interfering with efficient proton transfer. Since the artificial intelligence nowadays can predict accurate protein structure close to experimentally determined structure, my research question for the current research is, can we use AI to optimize protein structure to increase its activity by changing its dependence on a water molecule for proton transfer? The goal is to discover new protein sequences that can maintain the local geometry defined by residue glutanic 279, glutanic 289, arginine 286, and serine 319, and reduce the distance between glutanic 279 and cysteine 299 to be less than 3 angstrom. I hypothesize that varying sequences between residue 279 and residue 299 can generate protein structures that maintain the overall structure of BDP, while reducing the distance between glutanic 279 and cysteine 299. And it is expected to discover protein sequences that can reduce the distance between glutanic 279 and cysteine 299 to remove the dependency on a water molecule for the proton transfer. Now I will go over my method and procedure section. First, in order to reduce the number of enumerated sequences between glutanic 279 and cysteine 299, 20 different amino acids at 17 residue position were broken into 4 segments, and this reduced sequences to about 40,400. Python code was used to generate these 40,400 sequences for subsequent structure prediction. Second, the enumerated sequences were submitted to collect-fold batch for proton structure prediction. The outcome of this experiment is a pool of 3D structure files including coordinate information with with each atom in mutant sequences. Last, the 3D coordinates XYZ for 4 pairs of atoms were extracted, and the distance were calculated using the formula listed in the quad chart. Protein sequences yield all 4 distances were selected for future investigation. This analysis was done with Python code as well. The outcome of this experiment is a list of distance measurements for all 40,400 mutant sequences. The distance is one of the criteria to evaluate the success of this in the cyclical evolution experiment. Now looking at the results. The histogram of the radius of gyration value shown in figure 2 is the distribution of RG value for all predicted structure. The peak of this histogram indicates the most probable RG value observed in the datasets, which appears to be around 8.4 angstrom. The spread of the histogram, which extends from roughly 8.1 angstrom to 9.5 angstrom, reflects the variability in possible geometries sampled by enumerated sequences. The reference line at RG equals 8.06 angstrom, highlighted in red, is the RG value for the wild-type hydrogenous. The fact that the peak of the histogram is close to this reference line suggests that the most frequently observed RG value are near the proposed optimal value. From a biological perspective, this could imply that natural selection has favored a particular range of geometries that confer efficient catalytic function. Proteins with RG value near the reference line may be more effective catalysts, possibly due to optimal distance between catalytically important residues and an overall shape that is conducive to substrate binding and product release. The existence of configuration with RG value significantly different from the reference line may represent alternative conformations that could be less optimal for catalysis, or they might be intermediates in a dynamic process where the protein explore a range of conformations during its functional cycle. In summary, the histogram indicates a selection for a specific range of protein geometries. If the reference RG value correlates with optimal catalytic efficiency, the data suggests that the structural configuration around this RG value may have been evolutionarily optimized for catalytic function. This is an example of how physical properties such as the radius of gyration can reflect biological phenomena like natural selection and enzyme function. Figure 3 and 4 present an insightful depiction of an optimized PTP geometry, showcasing an adaptive structural modification that could enhance the catalytic efficiency of hydrogenase. Figure 3 illustrates a local PTP geometry where the proximity between glutanic 279 and cysteine 298 is significantly reduced. This configuration is postulated to diminish the dependence of the enzyme on the catalytic water molecule observed near the active site. The closer interaction between the two residues suggests a direct mechanisms of catalysis that bypasses the need for water-mediated reactions, potentially streamlining the enzymatic process. Figure 4 depicts the local structural changes due to specific mutations from phenylalanine 296 to glutanic, from threonine 297 to glutanic, and from serine 298 to cysteine. These mutations result in an aforementioned optimized geometry. The substitution of larger side chain residues with glycine, which has a smaller side chain, creates additional space that allows the remaining residue to reorient themselves closer together. The replacement of serine 298 with cysteine introduces a thiol group that could directly participate in the catalytic cycle, either through nucleophilic attack or by stabilizing transition states. These mutations collectively demonstrate a strategic redesign of the enzyme structure, which may lead to a more efficient catalytic pathway that is less dependent on water molecule, thereby potentially increasing the enzyme activity under a wider range of physiological conditions. This figure exemplifies the interplay between protein structure and function and highlights the potential of rational protein engineering in enhancing enzymatic activity. The implications of the results from figure 2 to 4 present a compelling case for utilization of artificial intelligence in evolution of enzyme design. The data suggests that the specific geometric configurations, as indicated by the distribution of Rg value, correlates with enhanced catalytic efficiency. AI can play a pivotal role in interpreting these findings and guiding the iterative process of enzyme optimization. AI algorithms, particularly machine learning and deep learning models, can analyze vast datasets of protein structure to identify patterns that contribute to high catalytic efficiency. In the case of the hydrogenase enzyme, the proximity of glutamic 279 and cysteine 298 and their impact on dependency on catalytic water molecule can be analyzed through AI to predict which mutations might result in a similar beneficial geometric changes. In conclusion, my research provides a framework for future in silico experiment using current AI technology to evolve protein function. The results will also provide a basis for future experimental tests of protein functions in a wet lab. The independence of hydrogenase function on this specific water molecule has a potential of increasing protein stability as well as increasing enzymatic efficiency, spurring the advancements of biological hydrogen production. Thank you so much for your time and interest in my research.